Hello, everyone, and welcome to today's webinar, Clinical and Operational Aspects of Measuring Hemoglobin A1c by Capillary Electrophoresis. I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Cebia. To learn more, visit them at cebia.com. Now, we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speaker, Dr. David Grenache, Chief Scientific Officer at TriCore Reference Laboratories. Dr. Grenache, welcome. You may now begin your presentation.
Well, thank you for the introduction. It's my pleasure to be here with you guys today to talk about some clinical and operational aspects of measuring hemoglobin A1c by capillary electrophoresis. Per my disclosures, I do acknowledge that I have received speaker honorariums from Cebia. And these are the learning objectives for my presentation today. All right, just so we can put my whole presentation in context, I want to tell you just a little bit about where I work at TriCore Reference Laboratories. We're an independent, nonprofit laboratory system located in the state of New Mexico, in the southwestern part of the United States. And we like to consider ourselves New Mexico's laboratory because we have our operation really across the entire state. We operate as a hub and spoke model. So we have one centralized core laboratory where I am in the city of Albuquerque. But TriCore also operates hospital laboratories in 18 hospitals and cancer centers around the state. And we do serve as the laboratory for the three largest healthcare systems in the state of New Mexico. We also have several branch labs. These are smaller laboratories that are located in urgent care centers. And we have 29 patient care centers where people can go and have a phlebotomy performed. Our menu consists of just about 3,000 different tests, and we perform approximately 1 million tests a month. We are CAP 15189 accredited, so all the usual things that you think about in a laboratory, but we are a system lab, right? So any tests that are not done in the hospital or cancer center laboratories, those laboratories do tests that are required for immediate patient care, and anything that's not urgent is brought here to the core laboratory. So let's get started. I'm going to start with a case report about a 36-year-old woman of Sicilian descent. So she has a history of obesity and hypertension, and she complains of being tired, and she has frequent urination at night. Her fasting plasma glucose is 124. You'll probably recognize that a diagnosis of diabetes mellitus requires one of the diagnostic criteria is a fasting plasma glucose of 126 or higher. So she's pretty close to that. Her hemoglobin A1c, as performed by an immunoassay, was normal at 5.3%. And her provider assured her that she did not have diabetes. But 4 months later, her fasting plasma glucose is 142, so it has crossed that diagnostic threshold. A 2-hour 75-gram oral glucose tolerance test was performed, and the 2-hour result was 220, which also exceeded the cutoff of less than 200 to establish a diagnosis of diabetes. And her A1c performed at that time was by high-performance liquid chromatography was 16.7%, so obviously extremely elevated. And notably, a presumptive hemoglobin variant was noted in her test result. She was eventually diagnosed with diabetes, but also with a hemoglobin S beta plus thalassemia. So that's clearly a little bit unusual, right? We have these two very discrepant hemoglobin A1c results performed over a 4-month period. We'll talk more about why that occurred later on. So let's just do a quick review of some basic things like hemoglobin. In most individuals, hemoglobins can be categorized into three groups. There's hemoglobin A, which represents about 97% of the hemoglobins, and then some minor hemoglobins. Hemoglobin F, very low, 0.5 or often 0%, and hemoglobin A2, about 2.5%. There are some there are some hemoglobin variants that are important. I have those off to the side, and we'll talk about those a little bit more later on. But I really want to point out here that the majority of hemoglobin A is not glycated, right? About 96-97% of hemoglobin A is not 94% is not glycated. But approximately 6% of hemoglobin A, which is referred to as hemoglobin A1, is glycated. And of those glycated hemoglobins, hemoglobin A1A and hemoglobin A1B make up about 1% of that glycated hemoglobin. And hemoglobin A1c, the star of today's show, makes up about 5% of hemoglobin A1. And those glycated hemoglobins include different subspecies. So, for example, if we look under the umbrella of hemoglobin A1s, the glycated hemoglobins, hemoglobin A1A1 is glycated with a molecule of fructose 1,6-diphosphate. Hemoglobin A1A2 is glycated with glucose 6-phosphate. Hemoglobin A1B has pyruvate attached to it. And hemoglobin A1c, again, the star of our show today, has a molecule of glucose attached to it. And as you're very well aware, hemoglobin A1c reflects the average blood glucose concentration over the previous 2 to 3 months, which is the average lifespan of a normal red blood cell. So hemoglobin A1c is currently the most widely used index of average glycemia for the routine monitoring and prevention of long-term complications of diabetes. And further, the American Diabetes Association and the World Health Organization also endorse the use of hemoglobin A1c for the diagnosis of diabetes mellitus. Because of those clinical uses, it's really important that we be aware of the factors that are known to interfere with hemoglobin A1c tests. And these interferences can be caused by specific analytical methods that are used to measure hemoglobin A1c, as well as through biological variables. So I mentioned that there are some abnormal hemoglobins, and I want to go into a brief explanation of the 4 most common ones, because these do contribute to some of that analytical and the biological sources of variation that we have to understand when we're selecting an A1c method and then interpreting results, because not all patients have just hemoglobin A. So these hemoglobinopathies, or these hemoglobin variants, arise from mutated forms of the genes that encode either the alpha globin or the beta globin chains of hemoglobin. And that results in some amino acid changes that create these abnormal hemoglobin variants. And while there are literally hundreds of hemoglobin variants that have been identified, the 4 most common ones that are mutations in the beta globin chain are shown here. So starting in the upper left-hand corner, we have hemoglobin S, right? Hemoglobin S is created when the amino acid valine is substituted for glutamic acid at the 6th amino acid position of the beta globin chain. And individuals can be heterozygous. They can have a phenotype of hemoglobin AS. These individuals are typically asymptomatic. Their red blood cells have a normal lifespan. But individuals who are homozygous, who have inherited 2 copies of the hemoglobin, of the mutation that causes hemoglobin S, have sickle cell anemia. And they have a decreased RBC lifespan, very short, about 15 days. Right underneath that, in the lower left-hand side, is hemoglobin C. Here, hemoglobin C is produced when there's a lysine amino acid substituted for the glutamic acid at the 6th amino acid position. Again, individuals who are heterozygous are asymptomatic, normal RBC lifespan. Homozygous individuals who have hemoglobin C disease have a very mild hemolytic anemia with a slightly decreased RBC lifespan. In the upper right, we have hemoglobin D. We have a substitution of glutamine for glutamic acid, but at the 121st amino acid position of the beta globin chain. And there are several variants of hemoglobin D, but the most common one is D Punjab. Heterozygous individuals, like the other variants, are asymptomatic with a normal RBC lifespan. Homozygous individuals do have a mild hemolytic anemia with a decreased RBC lifespan. And then hemoglobin E is a glutamic acid substituted for lysine at the 26th position. There is a slight reduction in these individuals. Hemoglobin E show a slight reduction in the synthesis of beta globin. Heterozygous individuals are asymptomatic with a normal lifespan of RBCs. Homozygous individuals have a mild microcytic anemia, and because of the decreased synthesis of the beta globin chain, they have indicators that suggest that give them a phenotype of beta thalassemia minor. So that's the last hemoglobinopathy I want to talk about, is beta thalassemias. So these mutations don't result in an amino acid substitution of the beta globin chain. These mutations decrease the synthesis of the globin chains. So we can have because there's a decreased synthesis of the beta globin chain. These individuals have increased synthesis of the delta chain. And so if we have hemoglobin A2 is composed of 2 alpha chains and 2 delta chains, and they have increased synthesis of the gamma chains, and so individuals who have hemoglobin F have a structure of hemoglobin that's 2 alpha chains and 2 gamma chains. Individuals who have beta thalassemia can be classified into either those 2 different groups. Those that are transfusion independent. We often refer to this as beta thalassemia minor because they have 1 gene that's mutated. They have a mild microcytic anemia, elevations usually of hemoglobin A2. And then beta thalassemia major, where you have both genes affected. These individuals have a microcytic anemia and they are often transfusion dependent. On the matrix on the right, what I'm showing you here are the clinical manifestations of both homozygous individuals and compound heterozygous individuals for the hemoglobin variants in beta thalassemia, because as that case report demonstrated at the start, we had an individual who had hemoglobin S beta thalassemia. And so you can see in the matrix, when individuals have these either homozygous conditions or compound heterozygous conditions, they often have some sort of decreased lifespan and of their red blood cells, and they have anemia. So with that as background, I want to talk about the different methods, the analytical methods that are available to measure hemoglobin A1c in clinical laboratories. At a recent so to give you an idea of the predominance of the different methods, I'm going to start with this slide. So when we look at recent CAP survey data, we can see that the most commonly used method to measure hemoglobin A1c is immunoassay. 51% of laboratories that are reporting utilize an immunoassay method. That's followed by HPLC, followed by an enzymatic method, followed by boronate affinity chromatography, and then followed lastly by capillary electrophoresis. So I want to talk a little bit about each one of these methods and some of their pros and cons. And we'll start with immunoassays. So most immunoassays are turbidimetric inhibition tests. And shown in the panel A, imagine a situation where there's no hemoglobin A1c present at all. So there's no blood present in the sample at all. There are reagent polyhaptenes these polyhaptenes are just synthetic molecules that contain multiple A1c epitopes. And they react with antibodies to hemoglobin A1c that are conjugated to a microparticle. And this combination of these polyhaptenes and these antibodies bound to a microparticle cause the microparticles to form an insoluble complex that precipitates and results in significant light scattering. Now imagine this situation where a blood sample is introduced and hemoglobin A1c is present. The hemoglobin A1c in the sample also reacts with those anti-hemoglobin A1c antibodies, forming complexes that reduce that. They bind up those antibodies, so it prevents the polyhaptenes from binding, and there's a reduction in light scattering. And what's actually measured is the rate of reaction. That reaction rate is measured turbidimetrically, and such that the light scattering, the amount of light scattering, is inversely proportional to the amount of A1c in the sample. I think it's important to point out that early immunoassays, so the first generation of A1c immunoassays, they tended to overestimate hemoglobin A1c in individuals who had either hemoglobin S or hemoglobin C traits. And that was because the antibodies in the first generation test spanned the epitopes that they detected, the epitopes that those antibodies detected, spanned amino acid number 6, which, as you remember, is the amino acid that is substituted in hemoglobin S and hemoglobin C. However, current immunoassays, so the ones that are in use today, do yield accurate results because the antibodies now span amino acids 1 to 4, so that it avoids that amino acid number 6. However, even though those immunoassays that are in use today span amino acids 1 to 4, they do yield accurate results, but they will also produce a result, a hemoglobin A1c result, in individuals who don't even have hemoglobin A. So, for example, individuals with hemoglobin S disease that are homozygous for hemoglobin S, they'll get an A1c result. And while that alone is misleading, right, because it's hard, you have a hemoglobin A1c result in a person who doesn't even have hemoglobin A, so that result alone is misleading. But also individuals with hemoglobin, with sickle cell disease, their red blood cells have a decreased lifespan. And if red blood cells have a decreased lifespan, then hemoglobin A1c results will be clinically misleading because they'll be lower than they would be in individuals with shortened RBC lifespans. And it's also important to note that an immunoassay will not be able to detect if an individual has a hemoglobin variant or not. And why that is important, I'll discuss a little bit later. The next assay I want to talk about is the enzymatic assays. And enzymatic assays utilize a protease to produce small peptides and amino acids from the beta globin chain, including the N-terminal glycated valines from the hemoglobin A1c molecule. That's shown up at the top on the left-hand side of that drawing. Enzymatic removal of the sugar residue from that glycated valine produces hydrogen peroxide. And the formation of hydrogen peroxide is measured using a colorimetric assay. And like immunoassays, enzymatic assays fail to detect if a hemoglobin variant is present, and so it will produce clinically misleading results in patients who don't have hemoglobin A. In boronate affinity chromatography, a lysate of red blood cells is added to a column that contains boronic acid. And at an alkaline pH, the glycated proteins are bound to the column, while the non-glycated proteins flow through the column. After that, an acid buffer is added to the column to release the bound glycated proteins. The beauty of this method is that it produces just 2 hemoglobin peaks, a non-glycated hemoglobin peak and a glycated hemoglobin peak, making the result interpretation relatively simple. However, these methods measure total glycated hemoglobin, not just hemoglobin A1c. And so the result has to be mathematically corrected to yield a hemoglobin A1c equivalent result. Boronate affinity chromatography methods are unaffected by the presence of hemoglobin variants, but they can produce clinically misleading results because, again, they don't detect hemoglobin variants, and again, because of shortened RBC lifespan in homozygous individuals, the A1C result can be clinically misleading. The second most common method for measuring hemoglobin A1c is HPLC. It's an ion exchange, high-performance liquid chromatography that separates the hemoglobin species based upon charge differences. So positively charged hemoglobin molecules move more slowly than the negatively charged molecules through the column because the column is packed with a negatively charged resin. So hemoglobin A1c will elute first because it's more negatively charged than non-glycated hemoglobin A. Other hemoglobin species and some hemoglobin variants can be resolved by HPLC and presumptively identified because they appear as additional peaks in the chromatogram. However, although HPLC can often identify the presence of some hemoglobin variants, it's important to note that different HPLC methods, even from the same vendor, have different resolving power, right? So the ability to separate those hemoglobin peaks from each other is can be really important, so that the method doesn't produce an inaccurate result. And lastly, the last method I want to talk about, which I'll go into a little bit more detail later on, is capillary electrophoresis. So capillary electrophoresis is a method that combines elements of classical zone electrophoresis with liquid chromatography. And while it's commonly used in laboratories for serum protein electrophoresis testing, it is less commonly used for A1C testing, with only about 2% of laboratories utilizing that method, as I showed you earlier. So capillary electrophoresis separates hemoglobin species based upon their electrophoretic mobility and electroosmotic flow in an alkaline buffer. After the sample is introduced at the anodic end of the capillary, a high voltage protein separation is performed, and direct detection of the hemoglobins is made at the cathodal end of the capillary. Direct detection right through the capillary using a detector and a wavelength of light at 415 nanometers. Now, like HPLC methods, capillary electrophoresis can detect the presence of hemoglobin variants, but the advantage of capillary electrophoresis is that it has extremely high resolution. And so we can completely separate hemoglobin variants from the other hemoglobins. And that results in a method that does not cause or is not susceptible to interference from other hemoglobin variants. And we can completely separate hemoglobin A from hemoglobin A1c. So I want to tell you now about the TriCore experience with hemoglobin A1c testing, and a little bit about our journey and where we've landed. So just to give you an idea of the testing volumes that we have, I have this graph, right? So we perform about 28,000 hemoglobin A1c tests every month here in our core laboratory. And the testing is performed in our hematology laboratory. And that's done because so often we get a single Labrador top tube for both a CBC and a hemoglobin A1c test. So it's a little bit more efficient for us to have to have A1c testing done in our hematology laboratory. If we go back a few decades to 1998, that's when TriCore started, and we began testing hemoglobin A1c using an HPLC system. And we used that system for several years. In 2012, we had to change our platform because our instruments were aging. And we were on an HPLC system for several years after that. But in 2019, we had to do an RFP to consider replacement options because our current instruments were aging and the columns had to be replaced frequently. We had a lot of service calls. It was just time to replace them. And rather than just automatically consider a replacement method that was HPLC, we decided to look at all of our options. So we looked at these four. We looked at two HPLC methods, that's the TOSO G8 and the Biorad D100. We also considered the boronate affinity chromatography method, the Trinity Premier method, and we considered capillary electrophoresis. We involved key operators, so our key technologists in the laboratory, in the hematology laboratory, in the process, and they helped lead us in the decision making. So here's a little bit about what we believe to be the pros and cons of the methods that we evaluated. So for the TOSO G8, ion exchange HPLC, an advantage is its broad measuring range, right? Can measure hemoglobin A1c from a low of 4% to a high of 18%. But we actually saw that there were several disadvantages. It has a relatively slow throughput at only 35 samples per hour. And at our volume of testing, that's not fast enough. The evaluation of the chromatogram, you have to look at it because there can be variance. It has to be done on a little paper roll that comes off of the instrument. So the chromatograms are on a paper. We personally, our process was when we identified individuals that had a hemoglobin variant, we would create an index card and keep it in a file folder, and that we kept track of those individuals because when they came back, we wanted to make sure we were producing accurate A1C results to the best of our ability. There were reports of interferences from common hemoglobin variants with this method. And at that time that we were doing our evaluation, we could not report a hemoglobin A1c result that was greater than 9% in the presence of a common hemoglobin variant. That has subsequently been resolved by the manufacturer with a software update. But when we were going through this process, that was still quite a burden for us to handle because we had to not report those results and send those samples out to our referral laboratory. We couldn't report an A1c result if there was a hemoglobin F concentration greater than 10%, and hemoglobin A1c is reported as a percent of total hemoglobin rather than a percent of hemoglobin A1c plus hemoglobin A. The other method we evaluated was the boronate affinity chromatography method. That also had a very broad measuring range. The nice thing about it is the ease of interpretation because you only get those two peaks, glycated hemoglobin and non-glycated hemoglobin. And an advantage was that there's no interference from common hemoglobin variants. But again, we thought the throughput was a little bit too slow for us at 45 samples per hour. And it measures total glycated hemoglobin, and A1c has to be mathematically derived. Although there's no interference from hemoglobin variants, the presence or absence of a hemoglobin variant cannot be identified by a boronate affinity chromatography method. So we thought that we considered that to be a disadvantage. The other HPLC method we looked at had a much higher throughput than the other one at about 80 samples an hour, so that would meet our needs. It had a very broad measuring range. And there was no interference from common hemoglobin variants, but it reports hemoglobin A1c as a percent of the total hemoglobin, not as a percent of the hemoglobin A1c plus hemoglobin A. And then lastly, we evaluated capillary electrophoresis from Sabia. It has a high throughput, high enough for us at 70 samples per hour, a broad measuring range, no interference from common hemoglobin variants. Hemoglobin A1c is reported, as is recommended, as a percent of hemoglobin A1c plus hemoglobin A. And what really sealed the deal for us was the superior resolution of hemoglobins from capillary electrophoresis. The disadvantage is that we have to use Sabia's QC products. We can't use a third-party vendor for their QC. So after we considered all of our options, we settled on the capillary electrophoresis method. Again, because it has superior resolution, it can detect hemoglobin variants very easily because of that superior resolution. There's also no interference with hemoglobin A1c measurement. It's very automated. It has automated mixing of the whole blood sample, and it has high throughput. It certainly has high enough throughput for our needs. And I'm showing this slide just to give you an example of the resolution, right? So here are 4 different individuals that are heterozygous for either hemoglobin S, C, D, or E. And you can see that in each of these cases, hemoglobin A1c, which is this smaller peak out here, is completely resolved from the other hemoglobins, including hemoglobin A. So you can still easily determine the hemoglobin A1c concentration, even in the presence of these abnormal hemoglobins. This is our current workflow. Now that we have switched to a capillary electrophoresis method. So samples are received here at the core lab and into our sample management department. They're received and they're accessioned. We have a robotic sorter that sorts the samples for the hematology lab to a specific location for hematology on the robotic sorter. And then lab assistants bring the hemoglobin A1c and CBC samples to our CBC instrumentation. We use a Sysmex platform for CBCs, and it's on an automation line. And then at the end of that automation line right here is a sorter on the Sysmex line that will then sort samples that need an A1c into a A1c rack. And then that is located right next to the capillary instruments, what are shown there in that bottom figure, that bottom picture. And they do have to be manually loaded onto the instrument for the analysis to occur. But once they're on the instrument, the workflow is automated. The vast majority of A1c results will auto-verify. What holds any sample with a hemoglobin variant is not auto-verified. It is held in the LIS and for manual review. The way we report results is probably very similar to how your laboratory reports them. These instruments are interfaced, and so the results flow right across through into our middleware, Data Innovations. There are rules that are based in the DI middleware that are triggered based upon results, and I'll talk about some of those momentarily. And then the results and those comments that are rule triggered are sent into our LIS, which is Sunquest. And then Sunquest sends that data, those results, on to our client's EMR. The elements that we report are obviously the hemoglobin A1c result. We also report estimated average glucose as it is calculated from hemoglobin A1c result. And then any comments that are appropriate, that are automatically added based upon the rules we've built in our middleware, those are also added to the result as well when appropriate. So I'm going to go through the different comments that we do have. So here's essentially the rules and the comment that gets appended, and then I'm also showing you the frequency with which we see these scenarios on the far right-hand side. And this frequencies are calculated from just under 100,000 A1c results that have been performed in the past, few months. So, scenario of an A1c that's less than 4%, that's below the measuring range of the capillary instrument. We report a result that is less than 4%. The estimated average glucose is reported as just less than 68. And then that's the comment that we attached. It says that we're detecting hemoglobin A1c, but we can't measure it accurately. And then we comment that extremely low values can be associated with conditions that alter the lifespan of the red blood cells. And then we suggest that serum fructosamine can be considered as an alternative method to evaluate glycemic control in such individuals. And that's very rare. That happens, we see that about 0.05% of the time. The opposite end of that spectrum, if the hemoglobin A1c exceeds the measuring range, we report a greater than result. Estimated average glucose is reported as greater than 427, and we do not add any comment. Again, pretty low frequency with which we see such extremely high A1c results. If a interfering hemoglobin variant is detected, right, because there's hundreds of hemoglobin variants, it's possible that some of them could interfere with the hemoglobin A1c measurement in capillary electrophoresis. We haven't encountered any yet. We would not report an A1c result. Therefore, we don't report an estimated average glucose. And we would comment that we detected a hemoglobin variant that interferes with the measurement. And then we suggest performing hemoglobin electrophoresis if clinically indicated. And then, again, serum fructosamine could be considered as an alternate method. We have not yet encountered this situation in the 2 years that we've been using capillary electrophoresis. We do detect non-interfering hemoglobin variants like those common ones, S, D, C, and E. We do report, because we can report a hemoglobin A1c result, and we know it's accurate. We know that it's not interfered with from the method we're using. We do report estimated average glucose, and we say that we are detecting a hemoglobin variant that does not interfere with hemoglobin A1c measurements, and we suggest performing hemoglobin electrophoresis if clinically indicated. And we see this relatively commonly, about 0.5% of the samples that we test. 1 out of 200 samples. If hemoglobin F is less than 24%, then we can still report a hemoglobin A1c result. We report an average glucose, and we say that hemoglobin F is present at a concentration that doesn't interfere, suggests performing hemoglobin electrophoresis if indicated. And again, something that's relatively infrequently encountered. Even less frequently encountered are samples with a very high hemoglobin F concentration, greater than 23%. We cannot report A1c, so we do not report average glucose, and we say exactly that hemoglobin F is present at a concentration that can cause a negative interference with hemoglobin A1c, and we suggest performing hemoglobin electrophoresis if needed, and serum fructosamine as an alternative method for evaluating glycemic control. If no hemoglobin A is present, so in individuals with, say, sickle cell disease, then we don't report an A1c result because there's no hemoglobin A. And we tell them that there's no hemoglobin A detected because there's a homozygous or hemizygous hemoglobin variant. And we remind them that in the absence of any hemoglobin A, there is no hemoglobin A1c to be reported. And then suggest hemoglobin electrophoresis if needed and serum fructosamine to evaluate glycemic control. Let's see. And then, if hemoglobin A2 is between 3 and 7.8%, we do report A1c, we do report average glucose, but we're prompting them in our comment that there is an elevated concentration of A2 that could indicate a beta thalassemia syndrome. We suggest performing hemoglobin electrophoresis if needed, and we comment that hemoglobin A1c could be decreased due to a shortened lifespan of the red blood cells, and serum fructosamine could be considered as an alternative. And that happens fairly frequently, about 0.5%, again, about 1 out of every 200 samples. So while numbers like 0.5% of samples seems really low, right? Individuals that have hemoglobin variant or perhaps beta thalassemia, while 0.5% sounds low, based upon our volumes, it's a pretty substantial absolute number of samples that we encounter here at TriCore. So what I'm showing you here is that same volume graph, day, week, month, year, but now I've adjusted it for the absolute number of samples that actually have a hemoglobin variant that we have detected. And at 2,000 patients a year where we're detecting a hemoglobin variant, I think is a value add and an important piece of clinical information that may not have been recognized. And it's another reason why I'm really pleased with our decision to go to capillary electrophoresis because of our ability to report hemoglobin A1c result in such patients, and the fact that we can do them. So that's all I have for you today. Just as a summary, analytical methods and biological factors can interfere with hemoglobin A1c measurements or the interpretation of the result. Notably, though, 75% of laboratories utilize A1c methods that cannot detect a hemoglobin variant or that produce an A1c result in the absence of any hemoglobin A at all. Detecting hemoglobin variants, while not the intended purpose of an A1c test, is important to avoid reporting misleading A1c results. HPLC and capillary electrophoresis A1c methods can detect hemoglobin variants, but it's capillary electrophoresis that really provides that superior resolution of the hemoglobin species and minimizes interferences. And with that, I'd be happy to take any questions. Thank you for your time.
Thank you, Dr. Grenache, for that informative presentation. We will now start the live Q&A portion of this webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some great questions coming in. Our first question: what ultimately led you, Dr. Grenache, to capillary electrophoresis for HbA1c testing?
Well, as I explained in the presentation, we did that RFP and we involved our technical leaders in the laboratory. And I think what ultimately sealed the deal for us was the superior resolution that capillary electrophoresis method provides us and its ability to detect hemoglobin variants.
Thank you so much. Now, when people hear the term electrophoresis, they don't think about operational efficiency. How does a reference lab the size of TriCore manage their HbA1c testing volume using capillary electrophoresis?
Yeah, so you're right. I mean, the terms electrophoresis doesn't really evoke high throughput methods, right? But technologies have changed quite a bit over the years, and the capillary electrophoresis method that we now use is pretty high throughput. I mean, it rivals that of HPLC methods. Probably doesn't rival that of immunoassay though. For us, the instrument that we use has 12 capillaries, and we have two instruments. So we have the ability to do 24 hemoglobin A1c assessments simultaneously. And given our volumes, which are fairly high, we're able to keep up with that workload. So again, for us, it was that combination of, I think, what we consider to be a superior analytical method, coupled with the throughput that we needed to meet our volumes that we have.
Thank you so much. Now, Dr. Grenache, in the case of homozygous variant, why not indicate to the clinician that the RBC lifespan is likely affected and no measure of HbA1c or glycated HB is useful?
Yeah, the person who asked that is very observant. And I recognize that as something we needed to start doing as I prepared this presentation. We have a similar comment when there's elevated hemoglobin A2, which is suggestive of a beta thalassemia. But we neglected to add that to our comment when there is a homozygous variant. So that will be remedied. I thank the questioner for pointing that out.
Thank you so much. And I want to thank our audience for these great questions that are coming in. Dr. Grenache, do you send out the HGBF or have an analyzer to run that?
Well, I'm not quite sure I understand the question. When hemoglobin F is detected in in a hemoglobin A1c measurement, we don't report that. As long as it's less than the threshold, if hemoglobin F is less than I think 23, then we can still report the A1c. If it's greater than that, then we usually will we indicate that hemoglobin electrophoresis might be warranted. And if the clinician orders hemoglobin electrophoresis, and hemoglobin F is present, then that's something that we do report quantitatively.
Thank you so much. Now, how do immunoassay methods compare with separation methods in regards to samples with elevated HbF?
Oh gosh, that's a good question. I'm not very familiar with the effect of hemoglobin F on immunoassays for A1c. Probably they're okay. I'm not speaking with any experience because I've actually never used a hemoglobin A1c method, but that was an immunoassay based method, so I'm not as familiar. But I'd hypothesize that there isn't interference from hemoglobin F, but I'm sure there are members in the audience who know far better than me. And if you do know the answer, I'd encourage you to put it in the question box.
Thank you so much. Now, our next audience member says, "Hi, I have a question for follow-up of patients for HbA1c where HB variants are detected on CE. Will the patients become their own reference range for diabetic follow-up? How do you communicate this info to the clinician?"
Could you read that again?
Sure. And it's number 13, Dr. Grenache. I have a question for a follow-up patient for HbA1c where HB variants are detected on CE. Will the patients become their own reference range for diabetic follow-up? And how do you communicate this info to the clinician?
No, if we detected hemoglobin variant by capillary electrophoresis, all we say is that there is a hemoglobin variant that does or does not interfere with the A1c measurement. The method is not approved to identify the hemoglobin variant to the clinician. So we say that there is a variant, and that hemoglobin electrophoresis could be considered if it's clinically indicated. But in the vast majority of hemoglobin variants the vast majority of hemoglobin A1c assessments in patients with the hemoglobin variant, assuming it's heterozygous, are accurate. So there's no need for each patient to become their own reference range. Serial testing, of course, is important in patients with diabetes to monitor glycemic control. And in that regard, longitudinal data, so historical data, is important. And that's the responsibility of the clinician to interpret the most recent hemoglobin A1c result in light of the patient's historical results.
Thank you so much. Now, do you use your capillary analyzers for other testing or just HA1C?
Yeah, so we have two dedicated capillary electrophoresis instruments that are in the hematology lab that are only used for hemoglobin A1c assessments. We also have the same instruments in our immunology lab where we perform serum protein electrophoresis, hemoglobin electrophoresis, immunotyping, et cetera. So we still use the same capillary electrophoresis technology, but on different instruments that we use for hemoglobin A1c. Those are just used for A1c.
It looks like we have time for a couple of more questions. This next audience member says, "Thank you so much for this wonderful presentation. What is the effect of sample hemolysis in case of CE report interpretation?"
Can you tell me what number that is?
20. We have such great questions coming in from our audience.
Well, if the sample I understand. Unless the sample is entirely hemolyzed, which is very unlikely, there shouldn't be an effect on the A1C result. We do want intact red blood cells, which are subsequently hemolyzed, right, as part of the procedure to release the hemoglobin from those red blood cells. But if there is some hemolysis already in the sample, it should not be impactful.
Thank you so much. We have time for one more question. Dr. Grenache, to follow up, immunoassay methods commonly stipulate that A1C should not be reported if the patient has elevated HbF over 7% or thereabouts, however, cannot detect for the presence of HbF. How could this negatively impact the results?
Thank you for the clarification. So remember earlier I had said I'm really not familiar with the impact of hemoglobin F on A1C measurements by immunoassay. So if this individual has shared with us that they commonly say that you should not report an A1C result if hemoglobin F is over 7%. That's a challenge. In an immunoassay method, you're not going to know the hemoglobin F percentage. So it's kind of nice that the immunoassay methods tell you that that's a problem, but the laboratory is not going to have any awareness of what the actual abundance of hemoglobin F is, because they're not going to be testing or looking for it. So that's a conundrum, right? I think that's another reason why I'm pretty pleased that we're not using an immunoassay method for A1C.
Dr. Grenache, I want to thank you for your presentation. Would you like to provide any closing remarks to our audience before we go?
No, thanks for the attention and your interest in the presentation. We have our journey with hemoglobin A1C testing has been pretty steady for many years until we made this switch. But the laboratory is very happy with the instruments and the method that we've chosen. And I'm really, I have a lot of confidence in the results that we report. And I really am pleased that we're able to identify patients who have hemoglobin variants, yet still, in nearly all cases we've encountered, still able to report an accurate A1C, with the caveat that in homozygous patients there is a decrease in there's no hemoglobin A, but there's the risk of decreased red blood cell lifespan and therefore an effect on the A1C result. So those are my final comments.
Well, I want to thank you again for your time and your presentation and for your important research. And I'd also like to thank Labroots and our sponsor, CBF, for underwriting today's educational webcast. Before we go, I also want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. So Labroots will alert you via email when it's available for replay. We encourage you to share that email with the colleagues who missed today's live event. Until next time, take care, everyone. Bye-bye.
Thank you.